Good afternoon. I'm Dr. David Pedler, the Scientific Director of the Canadian Institute for Military and Veteran Health Research, also known as CIMVR. Thank you for logging into today's Lunch and Learn. It's called Canadian Forces, Health Services, and Defence Research and Development Canada, Research in the Time of COVID-19, Pushing Through. Before we begin, let's acknowledge the Indigenous peoples of all the lands that we are on today. While we're meeting virtually, I would like to take a moment to acknowledge the importance of the land which we each call home. We do this to reaffirm our commitment and responsibility in improving relationships between nations and to improve our own understanding of local Indigenous peoples and their cultures. I will now invite the CIMVR Associate Scientific Director, Dr. Stephanie Belanger, to say a few words, as well as to introduce our moderator. Thank you. Stephanie?
Merci, David. Bonjour à tous. Au cours de cette séance, nous présenterons 4 collaborations de recherche qui ont traversé la pandémie et produit des résultats qui pourraient offrir des avantages cliniques au personnel des Forces armées canadiennes et à tous les Canadiens et Canadiennes. Tout au long de la séance d'apprentissage aujourd'hui, nous vous encourageons à poser des questions dans le bavardoir auxquelles nous pourrons répondre pendant la table ronde. Throughout today's lunch and learned, we encourage you to ask questions in the chat room that we can address and discuss during the panel discussion. It is now my esteemed pleasure to introduce today's moderator, Lieutenant Colonel Andrew Beckett. Lieutenant Colonel Beckett is the Trauma Advisor to the Surgeon General, Chief of General Surgery for the Canadian Armed Forces, Assistant Professor at both the University of Toronto and McGill University, and Medical Director of the Trauma Program at St. Michael's Hospital. Lieutenant Colonel Beckett.
Thanks, everybody, and thanks so much to CIMVR for hosting this event, and thanks to all of you for taking the time to join us today. You know, I think it's been a very challenging time for researchers in the defense community, and but I think we have a long history of pushing through difficulties in research over the decades. Many of you have heard of Banting and Best, the co-discoverers of insulin, and they were both Royal Canadian Army Medical Corps veterans of the First World War, and they learned how to do some of their research during those troublesome years of the First World War, and push through the times of the 1920s doing research in Toronto. And these Banting and Best also became very important researchers during the Second World War as well, with innovations like the G-suit and freeze-dried plasma. So, you know, the Canadian Forces and Defence Research Development Canada have a long history of being able to produce excellent research during these troublesome times, and the COVID-19 pandemic is no different. So today, we just wanted to give us some updates of our research in progress that we've been conducting over the course of the pandemic. And we think that it's going to be very meaningful to the Canadian Forces, the defense community, Canadians at large, and potentially global populations. So we've really lined up some excellent speakers, and I'd like to thank all the speakers in advance, Dana Devine from Canadian Blood Services, Christian Kastrup from the University of British Columbia, and Henry Peng from Defence Research Development Canada, and Michelle Schultzberg from the University of Toronto. So again, thanks to all the speakers, and I think we've got a really good lineup for you today to sample a little bit of what we've done, and I look forward to answering your questions during the discussion panel.
Okay, thank you very much for giving me the opportunity to tell you about our work developing counterflow for non-compressible inter-abdominal hemorrhage. I'm an associate professor at UBC. I'm also the chief scientific officer of Commotion Drug Delivery Systems. Before we get started, I really want to thank CIMVR and CFHS for giving me the opportunity and supporting our research over the years, in addition to DRDC. I really want to thank Lieutenant Colonel Andrew Beckett, as well as many others that have supported our work. So General Bilodeau and Roger Tremblay, and Colonel Lexi Natal, Colonel Clifford at DRDC, Stephen Bjarnson, of course Hugh Semple and Catherine Ten, as well as at CIMVR, David Pedlar and Stephanie Bellinger. So the major challenge we're working to address is non-compressible inter-abdominal hemorrhage. It's a major problem in military medicine. It's a common cause of preventable death on the battlefield. It's also responsible for 40% of civilian trauma deaths due to hemorrhage. It's challenging because there's often several bleeding sites in the abdomen. They're inaccessible, so you can't compress them, and it's hard to even find them and visualize them. What's really needed is immediate surgery, but this is challenging. It's challenging to get the patient to surgery before it's fatal, especially if there's delays in transport. This is a prolonged field care situation, and in future conflicts without air superiority, this is expected to be a major issue. Non-compressible hemorrhage can be quickly fatal without surgery. And in the data I'm showing here, this is in pigs and experiments we did at St. Michael's Hospital with Andrew Beckett, Hugh Semple, and others. And so in this swine model of non-compressible liver hemorrhage, what we see is that most of these animals die within an hour from this injury. And this recapitulates what's seen in people. And so if you can't get to surgery fast, these non-compressible bleeds in the abdomen are fatal. We wanted to come up with a way to address this. I want to give a quick overview of other approaches that are being developed or used for this. And these are non-surgical interventions. They include a REBOA, the abdominal aortic junctional tourniquet, as well as abdominal foams, such as a rescue foam that's in development. These are really promising, but these products can only be used for 1 or 2 hours before they cause major problems like distal ischemia and compartment syndrome. And with REBOA and rescue foam, it's challenging to administer them pre-hospital. So REBOA can be used by surgeons, but it's challenging for general practitioners and very challenging for a medic. And so I wanted to come up with a way to address this. So non-compressible bleeds are challenging, as I mentioned. Why not inject hemostatic agents right into the abdomen? So thrombin, other agents like tranexamic acid are used by surgeons to stop these bleeds. Why can't you just inject it into that compartment? The answer that's been developed over the many decades is that hemostatic agents are not effective because you put that agent into the abdomen, it can't be transported to the bleeding site to stop that bleed. Okay, so I'll tell you about the work that we're doing to overcome this challenge, and I'll show you data in an external bleed, such as femoral artery bleeds, and then we'll show you what we're doing more recently in the abdomen. So to overcome this transport problem is critical. And so in the schematic, showing an intractable case, it's not good, and a manageable case. In the intractable case, as you can see in the red, this blood flow is pushing this external hemostatic agent away from this. And so we wanted to come up with a way to keep that hemostatic agent deep into the wound, to push it in the wound and generate some compression. So keeping the hemostatic agent in with compression is possible, but without compression, this is a major challenge. And that's what's seen in this data. So this is a femoral artery injury in pigs, looking at the amount of pigs that survived for 3 hours. This is done in collaboration with Nathan White at the University of Washington. And so what we see is if we don't compress these wounds, even with the standards like combat gauze and thrombin on gauze, so thrombin on gauze is used during surgery, most of these pigs will die within 3 hours. And so compression is not always possible in the abdomen, as well as for these external bleeds, such as the femoral artery injury, if it's a care under fire situation, or if it's a self-care or buddy care, compression can be challenging. So what can we do to address this? So we developed a hemostatic agent that could, an agent that could help spread the hemostatic throughout the wound. And so we've named it counterflow. Pretty simplistic chemistry. We take calcium carbonate microparticles, that's what's shown here, and an organic acid. In our case, we use tranexamic acid, which is a clot stabilizing agent. We mix these together and put it into blood. It releases gas. It releases CO2 that effervesces, spreads the agent throughout the wound, and also generates some pressure from the CO2. And calcium carbonate and CO2, these are very safe. So they're bioresorbable. CO2 is very soluble in blood. It's used during laparoscopic procedures routinely. So it's a very, very safe approach. And so I'll show you what happens if we just take this simple, simple chemistry and mix it with thrombin gauze. And so right here, most of the pigs died. When we take when we add counterflow agent to it, it takes the thrombin and tranexamic acid and makes it much more effective. And all 8 of the pigs lived in this experiment. And we've repeated this data with Andrew Beckett and with Hugh Semple, and always seen top efficacy with counterflow on gauze. And so this is just a picture of the gauze in a femoral artery injury, and so you can see the gauze packed into this wound. It's releasing a lot of CO2 to help with compression, but it's also delivering systemic tranexamic acid. It's delivering calcium and thrombin is optional. Works better with thrombin, but works without thrombin in many cases also. And so this work has been supported extensively by DRDC as well as the Surgeon General's Health Research Program, and so we're really appreciative for that and really looking forward to continuing this. So at this point, we're doing moving to GMP manufacturing, further safety and efficacy, and we hope to be using this in people in just a couple of years, working together with many people on this call. So the hypothesis we're currently testing is that by enhancing the transport of hemostatic agents in wounds, that we can manage abdominal hemorrhage, this big unmet need. And so to start, we developed a way to spray the powder. And so we started in a more easier to reach bleed. And so what we did is we threw down through the esophagus and a gastroscope, we put our powder into a vise and sprayed it through a catheter into a bleed in the stomach. In this case, there were arteries in the abdomen that were surgically placed into the stomach, and those are the arteries that we damaged. You can see it's a high velocity pulsatile bleed, and we can stop these very well. So within just a few minutes, we can stop these bleeds with just a couple grams of powder. And this no rebleeding occurs in these pigs. We kept them alive for 7 days. And so this was published just recently with Drs. Beckett and Semple, and then a gastroenterologist, Fergal Donnellan. And so really what we're trying to get after, non-compressible inter-abdominal hemorrhage. And so this is the experiment that we've been doing more recently with funding administered by CIMBER. And so in a swine model, we make an injury to the liver, and then we spray our powder through this device. There's just a CO2 cartridge here, and you can see the powder coming out. And so an optional thing we can do is do ultrasound to find this bleed. You don't have to use ultrasound in the real setting. So, and then we do is we place the catheter, and this is a Sellinger technique. This is something that a general practitioner can easily do. We think that medics could be trained to do this, and it doesn't require a large injury to be made like other like rescue foam, for example. And so we spray the powder through this catheter and to address these injuries. And so first we do we remove a portion of the blood to make the pigs to bring the pigs into a challenging state. We do a liver, remove a piece of the liver, and then we either give them a gram of a TXA with lactated ringers, or in addition to that, we administer counterflow and monitor thereafter. And so in the first, this was just done in the last couple months, in the first set of data, we're really excited. What we're able to do is in animals that had severe hemorrhage, where their mean arterial pressure dropped below 35 millimeters of mercury, we're able to drastically change the survival of these animals. So most of the animals, the mean survival without counterflow was 0.8 hours, and then with counterflow, we were able to take the median survival and push that out to the 2.4 hours. So really happy to be able to extend this this time frame, and we hope with a continued development we can extend this even further. And it's created, why does this work? It does create, the CO2 creates some pressure in the abdomen, but it keeps it in a safe level, the same level you'd see during a laparoscopic surgery. And so it's a safe but useful CO2 pressure. And also it makes very robust clots. And so it's just a picture of how stretching this clot out, how robust it is. So I'm sorry for the gross picture, but I just want to emphasize that these clots are more like organs at this point than they are a normal clot. So they're very stiff. And we really think that the counterflow, the gas being released, is pushing the agent throughout the wound to stop the bleed. And so in conclusion, non-compressible inter-abdominal hemorrhage is a major problem in military medicine. The current devices have major limitations, but I think counterflow is a promising new approach towards this. It can be administered to the abdomen without surgery. And yeah, in our first attempt at extended survival in non-compressible inter-abdominal hemorrhage in swine. So really excited about this, and really excited to keep collaborating with many of you on this, working and collaborating with many of you on this call as we go forward towards developing this for humans. And so, yeah, I want to thank the people in my lab that have done the work, Massimo, Nabil, and James, as well as these collaborators and these funding mechanisms of funding. And I really appreciate, again, the support that many of you have given to this. So thank you very much.
Well, thanks very much, Christian. That was great. And, you know, that's one of the big upcoming problems for future of battlefield medicine and combat casualty care is extending that golden hour into the golden 2 hours, the golden 3 hours, is to push that timeline to ensure we can get our patients in satisfactory condition to medical treatment facilities by extending that golden hour. So our next talk is from Dr. Dana Devine. She's the Chief Scientific Officer of Canadian Blood Services and very respected in the transfusion medicine and hematology research circles. And she's going to be talking to us on the production of freeze-dried plasma and also our program looking at convalescent freeze-dried plasma. And just for those of you starting to think of questions, please put them in the chat box and we'll triage the questions and make sure that we can get some answers for you during the panel discussion.
Okay, hello everyone. My name is Dana Devine. I am the Chief Scientist at Canadian Blood Services, and I was asked to share with you today some data and information that we have about the application of a relatively new technology in Canada, which is freeze drying of plasma to the preservation of COVID-19 convalescent plasma. So I'm very pleased to be able to do that today. I do have a couple of disclosures that are here. I have some interaction with some companies in the private sector. I'm the chair of the scientific advisory board of Mako Pharma, which makes most of the blood bags that are sold in Canada. And I also belong to the board of directors of a startup company called Storm Bio. In the last 4 years, I've received some research funding from the companies that are listed here. So I wanted to just say a few words about convalescent plasma, just to set the stage for you. So convalescent plasma is not a new therapy. We've been using convalescent plasma for a very long time. And these are photos here coming from the 1918 influenza outbreak. So we actually were attempting to use, well, not me personally, obviously, in 1918, but there was an effort made to use convalescent plasma to treat people who had come down with Spanish flu. And people had gone back about 10, 15 years ago now, and actually dug out all the old literature from the 1918 to 1925 period. And what they were able to determine in that literature was that if you got the convalescent plasma into patients quite early in their disease, that you could actually lower the fatality rate. And so this has led to a lot of interest in convalescent plasma in various epidemics over the following subsequent 100 years or so. We also had an opportunity to look at convalescent plasma in the SARS-1 outbreak in 2003, but there's been really quite a big focus on the use of this plasma in the current pandemic. And there were a lot of us running around a year ago setting up clinical trials and trying to understand whether COVID plasma would actually work in patients. And most of those trials actually failed, and I'll explain to you a little bit about why. And here's one of the trials that didn't fail. So this is a trial demonstrating that if you treated elderly patients with high titer plasma containing lots of antivirus antibody, and you got it into them early, that you could actually prevent severe respiratory disease. So you can see here that in the patients who got a placebo, severe respiratory disease came up in about 30% of people that got infected. And you could essentially cut that in half by giving them COVID convalescent plasma very early in their disease course. And so it became quite clear that treating patients early was really important. And that was one of the things that most of the clinical trials that have been set up around the world were not doing. And so what we know at this stage of the various clinical trials and other sorts of investigations that have been done are two really important things. One is that the plasma must have a sufficiently high titer of antibodies to be effective in the person that you're giving it to. And that the best way to assess this is to actually measure the amount of antibody that will neutralize live virus, because that's effectively what you're asking that antibody to do once you put it into somebody. And then the other important aspect is that really you have to get this convalescent plasma into patients before they start to mount their own immune response. And once they've got their own immune response going, adding a few more antibodies from a donor isn't really going to help. So these are the two things that have been our take-home lessons so far from all of the many clinical trials that have been done around the world looking at the use of COVID convalescent plasma in this pandemic. Well, one of the things that struck us was that if COVID convalescent plasma turns out to be effective for early use, that we want to be able to collect it from people who are just recovering from COVID convalescent plasma now, because that's when their antibody levels are the highest. And so you don't want to wait until somebody's a year past their COVID event, because their antibody level will probably have fallen back down to almost undetectable. And so what we had been doing was collecting COVID convalescent plasmas from people who were at least 28 days past the resolution of their symptoms, and then testing them on a viral neutralization assay, which is what is shown in this photograph here. We wanted to make sure that if you diluted the plasma, that your plasma was going to be able to kill virus at least 1 in 80 dilution, and preferably higher than that. So in the clinical trials, we were using 1 in 160, but for these particular studies, we had taken the plasmas that were donated that were testing a little bit lower than that. And the way that we do these is we grow up a live SARS-CoV-2 virus in viro cell cultures, which is a monkey kidney cell. And then we actually put the plasma on there and look to see how good the plasma is at killing the virus and preventing the formation of viral plaques in these assays. So once we actually had these products in hand, we were preparing pools of 10 of them and looking at what happened to the antibody reactivity. Once you pool them and split them back out and lyophilize them, they look like this, and you can reconstitute them with sterile water in about a minute. What we were able to show for the amount of effect of the treatment on the antibodies was that there really was very little effect. So when we looked in the 3 different kinds of ELISA assays shown here, so anti-RBD is the receptor binding domain, the anti-NPIgG, and then the anti-spike. So spike is the protein that interacts with the cell that the virus is trying to infect. And so we could show in all of these assays that we saw no significant difference before and after lyophilization. So this was really good. This meant that the active ingredient in the antibodies was still present. And as you can see in the little red box at the bottom, when we went and redid those plaque neutralization tests, we could also show that those assays were not affected by the freeze drying process. So this was good news. It meant that we could actually freeze dry plasma from COVID convalescent patients and be able to retain the function of the antibody. And then the other pieces of work we did were really to look at the effect of the freeze drying on complement and coagulation factor levels. And you'll hear a bit more about the function of the coagulation factors, but I just wanted to tell you about the actual factor level measurements that we made. We first of all looked at the complement system, which is a collection of molecules that bind to antibodies and are important in the actual killing of viral particles. We looked to see what the effect of the freeze drying was on the complement in these convalescent plasma products, and what we could show was that there was only a slight increase in the amount of complement activation, and it was very consistent with what we had seen when we made lyophilized plasma from fresh frozen plasma collected from people who never had COVID, so work we had done before the pandemic. And then similarly, when we looked at individual coagulation factor activity, we did see a small decrease, so decreasing in the activity levels of between 2 and 10% after lyophilization, with the exception of factor 8, which we already knew it doesn't really like to be lyophilized, and that had decreased by 18%. We looked at the prothrombin and activated partial thromboplastin times and could show that they were slightly pulled out after lyophilization, but this is really just a reflection of this 2 to 10% impact on a lot of different coagulation factors caused by the lyophilization. So this all looked very good. It looked like these plasmas were going to behave the way we would normally expect plasma to behave. So I'm going to stop talking and let us hear more about the information concerning the coagulation function that some of our colleagues are going to talk about. Thank you.
Thanks so much, Jana. That was great. And you know, one thing that we have thought about of using the freeze drying process to conserve convalescent plasma is not important just for COVID-19, but it could be important for other pandemics or even CBRN, biological warfare agents, as a countermeasure. So that's a really important thing that has come out of this, is a stable way of conserving that as a potential countermeasure. So the next talk is on to more freeze-dried plasma, which is one of the highest priorities within the NATO research group, is to have you know, every country being self-sufficient in terms of freeze-dried plasma. And my colleague, Dr. Henry Peng from DRDC, he's going to be talking about his work in looking at the coagulation profile of the freeze-dried plasma product that's been developed in conjunction with Canadian Blood Services and a company called Trumo BCT. So he's going to talk about all what we found in terms of how good this product is for coagulation in the pre-hospital environment.
Good afternoon. My name is Henry Peng. I'm a defined scientist at DRDC. I will talk about some of our work on freeze-dried plasma, from casualty care to COVID-19 therapy. The work was built on the long history of collaborations between CFHS and DRDC on translational trials in casualty care. It started 20 years ago with a single-center trial of hypertonic saline at Sunnybrook Hospital. Over the years, the collaboration has led to several high-profile multi-center trials, such as ROCK trial for pre-hospital hypertonic resuscitation and PURPLE trial for optimum blood product ratio in massive transfusion. Recently, we completed the first trial to evaluate the early use of fibrinogen concentrate in severe trauma, which has led to a multi-center trial of clotting factor concentrates being conducted in 10 hospitals. Despite of advances in casualty care, hemorrhage remains the leading cause of preventable death in the battlefield. Remote damage control resuscitation with blood products is recommended as early as possible. As a result, dry blood products are particularly useful for combat trauma in the battlefield, given their logistic advantages over liquid blood products. Studies have shown that freeze-dried plasma is stable for more than 10-15 months, even under field conditions, and can be easily administered in the battlefield without cold chain and thawing device, as is the case for fresh frozen plasma. There are currently 3 freeze-dried plasma products, or FDP, on the market. The French and German products have been used by military in pre-hospital settings, but they won't be approved by Health Canada due to the risk of prion disease transmission. In collaboration with Canadian Blood Services and DRDC, CFHS has produced Canadian freeze-dried plasma using the freeze-drying system from Tomorrow BCT company. The diagram shows the production process. Briefly, each unit of FDP is manufactured from Canadian sourced plasma collected from 10 healthy donors in a plastic bag. The product can be reconstituted in water in less than 2 minutes for transfusion. As it is produced in a single bag, there's no glass bottle breakage. The product is more rugged and suitable for remote or stereo settings. While we were working on the project, we were hit by the global pandemic, which has caused millions of deaths up to date. In Canada, we are in the 3rd wave with thousands of daily infections. There have been tremendous global efforts to find an effective treatment for COVID-19. As you have heard from Dana's presentation, convalescent plasma has drawn lots of attention and been widely studied. Built on our initial work on Canadian freeze-dried plasma, we are developing COVID-19 convalescent freeze-dried plasma, or CCFDP. Here are some advantages of CCFDP over the current convalescent plasma used in clinical trials. The first is the long stability, which would allow for stockpile and easy use in developing countries and deploy troops during future pandemics. Secondly, as this is a pooled plasma product of 10 donors, it would standardize neutralizing antibody activity compared to single donor convalescent plasma. Thirdly, CCFDP can be reconstituted and transfused in a smaller volume to get a higher antibody level and avoid volume overload. Here are some lab capabilities we use to characterize both Canadian FDP and convalescent FDP. First, we use a hemostatic analyzer called ROTEM to measure global hemostasis from the beginning with coagulation to the end with fibrolysis. We also used staggered coagulation analyzer to look at specific factors in coagulation pathways. The multiplex immunoassay system was used to measure immunological profiles as well as anti-SARS antibody level and various neutralizing activity. Because of the time limit, I will only highlight some of our main findings. The slide shows hemostatic and immunological profiles of 3 preparations of FDP reconstituted in water and their initial fresh frozen plasma. First, there were no differences in hemostatic variables for coagulation and fibrolysis, as well as inflammatory cytokine concentrations, suggesting the hemostatic and immunological proteins were preserved during the freeze drying process. In addition, there were no differences among the 3 preparations of reconstituted FDP, which are the samples immediately frozen at -80 or first stored at 4 degrees for 24 hours before being frozen at -80, or stored at room temperature for 4 hours before being frozen at -80, confirming short-term stability of FDP after reconstitution. This slide shows the antibody IgG concentrations as measured against each of 3 virus antigens and percentage inhibition of host receptor binding to the virus spike protein by the antibodies. The latter is the main mechanism by which the virus infects humans and considered as pseudo-neutralizing activity. First, we found both convalescent frozen plasma and its corresponding CCFDP showed 2 orders of magnitude higher antibody levels and more than 4 times higher binding inhibition than their pre-COVID healthy control. In addition, there were no differences in either antibody level or neutralizing activity between convalescent frozen plasma and its correspondent CCFDP, suggesting the antibodies were preserved during the freeze-drying process. In summary, we have confirmed both Canadian FDP and convalescent FDP were equivalent to their respective fresh frozen plasma sources, with several potential logistic and therapeutic advantages. Clinical trials need to be conducted to evaluate their safety and efficacy for both civilian and military use. In terms of next steps, we'll continue our laboratory study of clinical trial samples from COVID patients. Health Canada has invited our submission for a clinical trial of Canadian freeze-dried plasma in bleeding patients. We will publish our scientific findings in open literature and DRDC reports. It has been a difficult time, but we are all in this together and supporting each other. As a silver lining, our collaboration with CFHS has become even stronger. With that, I'm looking forward to more discussion through our technical panel. Thank you very much.
Thanks so much, Henry. And this is a great example of the collaboration between Canadian Blood Services, Canadian Forces Health Services, CIMVAR, and DRDC on this very important project. And I like to think this is a very Canadian project. And in fact, in Canada during the Second World War, the 430,000 units of freeze-dried plasma were produced for the Allied war effort in making Canada the biggest producer of freeze-dried plasma for the Second World War, which was really important in the treatment of burns in England from RCAF pilots during the conflict. And so I like to think this is a continuation of this type of collaboration. So our last speaker is Michelle Schulzberg. She's a hematologist at St. Michael's Hospital, and she's going to talk to us about really getting involved in a clinical study, world-class clinical study looking at COVID-19 patients and how to improve their outcomes. And just as a reminder, please put your questions in the chat and we'll make sure we get those answered during the discussion panel.
It's a great honor and privilege to present to you all today on the RAPID trial, which is a pragmatic, frequentist, adaptive, randomized control trial that evaluates therapeutic versus prophylactic standard care anticoagulation as a rapid response to the COVID pandemic. Hospitalized patients with COVID-19 very often have laboratory evidence that is suggestive of coagulopathy, and this was found very early on in the pandemic. In fact, up to 70% of hospitalized patients have evidence of this on their blood work. And it's the D dimer, which is an indicator of both formation and lysis. That is what most characterizes this coagulopathy. And also this biomarker seems to correlate well with the need for critical care and death. Thus, the D-dimer is a very effective biomarker that can assist with prognostication of patients. Thromboinflammation does seem to be a key pathophysiologic mechanism of COVID-19 and as it progresses through various stages of severity. And from early on in the pandemic, we hypothesized that it might be important to intervene early with antithrombotic therapy, i.e. at stage 1, where the coagulopathy has not yet become disseminated. And this hypothesis came from an early observation out of Wuhan, China, that indicated that there was a lower 28-day mortality among patients who received heparin-based prophylaxis compared to those who did not, who had COVID-19. And this seemed to be particularly true amongst those who had a high D-dimer. But we were interested in the value of heparin beyond its anticoagulant effect. And this is because there are numerous animal models of acute lung injury and sepsis that describe the benefit of heparin, as it does seem to have a beneficial effect on endothelial function, capillary leak, and seems to improve acute lung injury. But also because heparin seems to bind directly to the SARS-CoV-2 spike protein, interfering with viral attachment and entry, and also because the heparins in general, both unfractionated and low molecular weight heparins, seem to have anti-inflammatory effects in acutely ill medical patients, and that includes those with COVID-19. So it was found that those with COVID-19 who were treated with low molecular weight heparin had lower IL-6 levels compared to those who did not. So current guidelines from the American Society of Hematology for inpatients who are moderately ill suggest prophylactic over intermediate or therapeutic intensity anticoagulation in those who are admitted to hospital who do not have suspected or confirmed thrombosis. And there's an encouragement for ongoing participation in clinical trials and observational epidemiologic studies while awaiting access to complete and peer-reviewed data. And so what do we know thus far about the management of patients who are admitted to hospital with COVID-19? So what kind of data do we have access to at this time? And so there's a preprint available that has come from the multi-platform trial of Active4A, ATTACK, and REMAP-CAP. This is a multi-platform adaptive trial where that indicated that there was no benefit to therapeutic versus prophylactic anticoagulation when provided to those who were critically ill. And in fact, there also seemed to be some indication for harm. And this was then replicated by the Inspiration investigators. This was recently published in JAMA, that compared prophylactic to intermediate dose heparins in the critically ill. Again, found no benefit and a signal for potential increased harm. And so as it said, it seems as if in the critically ill that therapeutic or even intermediate intensity heparin-based therapy is not of benefit with possibility for harm. But what do we know about the patients before they end up in the ICU? So the moderately ill or ward patients. So this data here also comes from the multi-platform trial of Active4, ATTACK, and REMAP-CAP. And here they're looking at ward patients. This data comes from a publicly available slide deck. And again, just like their critically ill randomized controlled trial, the primary outcome, again, is organ support free days alive to day 21. And the outcome of organ support free days is an ordinal scale that includes both in-hospital mortality and organ support free days. And they've defined organ support as ICU level care with the receipt of invasive or non-invasive mechanical ventilation, vasopressors, inotropes, ECMO, and also high flow nasal cannula oxygen. And so here is a pictorial representation of their preliminary findings. Of note, this isn't from a locked database, it's not peer reviewed. But you can see here, if you look at the scale above, so I'll just orient you, red is bad. It means death or greater need for organ support, and blue or increasingly blue means less organ support is required. And so this top chart over here, this is the prophylactic group compared to the therapeutic group. Importantly, in this multi-platform trial, the prophylactic group may have also included individuals who are on intermediate dose anticoagulation, as it was pragmatically defined as standard care, or what was standardly defined, or sorry, what was defined locally rather as standard prophylactic care, which may also have included escalated dose therapy, as this was adopted by many institutions early on in the pandemic. So it compares the standard care arm to the therapeutic arm. The top duo here is looking at patients with positive D-dimer. This here in the middle is looking at patients with low D-dimer, and over here are those where there was missing D-dimer data at the time that they revealed these slides. But overall, importantly, they found that there was a lower requirement for organ support, i.e. 16% compared to 23% when you compare therapeutic to standard care in their trial. So this is every scientist's dream to have your hypothesis come true. And so again, just to summarize the findings of the multi-platform trial. So for the moderate state, i.e. ward patients, they stopped their trial early as, and it was an adaptive trial, so they did conditional power analyses, et cetera, that indicated that there was a superiority versus the severe state, the critically ill. They stopped it early due to futility. So it seems as if higher intensity anticoagulation with heparin-based therapy is of benefit when provided to ward patients, and it doesn't seem to be of any benefit and possibly a risk of harm amongst those who are severely ill. So what do we do with those who are started on therapeutic and then get transferred to the ICUs? We don't know the answer to that question. So now I'm going to talk about our clinical trial, which is the Rapid COVID Coag trial, or the RAPID trial. So this is a two-arm parallel, pragmatic, frequentist, but also with some adaptive components, multi-center, open-label, randomized controlled trial, again, of moderately ill patients, ward patients, again, with the goal of starting heparin as early on in the patient's hospital admission for COVID-19, with the goal of decreasing the sequelae related to thromboinflammation. And it compares here prophylactic to therapeutic anticoagulation, but we were very strict in the definition of prophylactic. And the primary outcome is a composite of ICU admission, non-invasive and invasive, mechanical ventilation, or death at 28 days. So from an eligibility perspective, we're looking at patients with confirmed COVID-19 admitted to hospital specifically for COVID-19, and those who have a positive D-dimer. And we defined a positive D-dimer in one of two ways, either a D-dimer that was greater than or equal to 2 times the upper limit of normal, or simply a positive D-dimer above the upper limit of normal defined locally with an oxygen saturation of less than or equal to 93% on room air. We're also looking at adults, and we excluded individuals with an absolute indication or contraindication for therapeutic anticoagulation. And so here, looking at the therapeutic versus the standard care arm. So the therapeutic arm includes all types of heparins. Again, this was to facilitate pragmatism and the possibility for drug shortages in the middle of the pandemic. And the standard arm, again, is comparing these agents, but here we have strict dose capping based on the best available evidence as well as expert opinion. And so we dose capped based on both body mass index and creatinine clearance. So I described the primary composite outcome. Our key secondary outcomes are as follows, with safety outcomes pertaining to major bleeding according to the ISTH, as well as heparin-induced thrombocytopenia. And we're also, of course, interested in venous or arterial thromboembolic events. So sample size consideration is 462 patients were estimated to be required to detect a 15% risk difference from 50% in the control group to 35% in the experimental group. And we defined this sample size based on early data in the pandemic. Obviously, as the pandemic has worn on, our knowledge has evolved and changed, but this was from data coming out of Wuhan, China. We have now completed recruitment and have reached 465 participants. You may wonder why we went 3 participants over. That was just in the middle while we were awaiting the deliberation of our data safety monitoring board. And they were reviewing both the interim data as well as the conditional power analysis. And so we had approval from Health Canada to slightly exceed the sample size as we were awaiting their deliberation. And so the adaptive component of our trial was the conditional power analysis. And so essentially our data safety monitoring board not only evaluated interim data at certain set points in the trial that were defined a priori, but they also reviewed conditional power analysis to help us determine if we needed to decrease or increase our sample size as we went on, because we acknowledged that knowledge was rapidly evolving and that things could change. So statistical methods, quite straightforward and frequentist in approach. Intention to treat with a chi-square test for the main analysis of the primary outcome, with generalized linear regression model with binary distribution and identity link to derive a risk difference with 95% confidence intervals, with subgroup analyses for age, sex, time from symptom onset, corticosteroid administration, diabetes mellitus, cardiovascular disease, race, ethnicity, level of D-dimer, and degree of hypoxia. This is an international trial that includes sites from Canada, the United States, Europe, the University College Dublin in Ireland, Brazil, Saudi Arabia, and the United Arab Emirates. Here are the images of our rapid investigators as well as our research coordinators. I wanted to emphasize the grassroots nature of this clinical trial in that many of the sites participated with limited research funding and donated research infrastructure in kind, as they believed that this was such an important clinical trial and wanted to help us reach the answer as quickly as possible. So the significance of RAPID, as well as other clinical trials in this space, is the importance of repurposing readily available, affordable, well-understood therapies, because we've been using heparins for the last 70+ years, to potentially address a key pathophysiologic mechanism that may in fact also be complementary to another agent that falls in this class with those same characteristics, i.e. corticosteroids. So our trial, along with other clinical trials in this space, has the potential to immediately impact clinical care of patients with COVID-19 admitted to hospital wards internationally, irrespective of our findings, as we believe that randomized care of agents with sound biological plausibility rather than haphazard best guess care is what is most critically important, even in the middle of this difficult, unrelenting pandemic. And this will lead to subsequent patients receiving evidence-based therapies and spared from unnecessary harm. You may wonder why I'm not presenting any data today, as I've already indicated that we have completed recruitment. And that is because our last participants will complete their 28-day follow-up on May 10th. And so I remain blinded, just as everyone else, to the results of our clinical trial. The only individuals who are aware of our findings at this point, and in fact, they were just our interim findings, are our data safety monitoring board. And so I'm eagerly awaiting the results of our trial. And I really do strongly hope that our results will support those of the multi-platform trial for the moderately ill, as it would be wonderful to have another agent that we could add to our armamentarium to help diminish the severity of COVID-19. So this is the wonderful team of individuals that I've been working with. My co-principal investigators include Peter Uni as well as Mary Cushman, and a wonderful steering committee that we could not do this wonderful work without. And also, of course, our patient participants. Without them, we would have nothing. And then finally, I really wanted to thank our funders. So critically, the Department of National Defense, the DRDC, have just been outstanding in their support. So a huge, huge thanks. Without the support, we could not have conducted this trial with the speed that we did and with the accuracy and quality that we have. Also, thank you to St. Michael's Hospital Foundation, as well as St. Joseph's Health Center Foundation, who provided substantial funding to allow for the trial to move forward, as well as funding, as well as research infrastructure in kind that was donated by many sites around the world who are involved in the RAPID trial. And so I thank you all very much, and I'm happy to answer any questions. Thanks.
Thanks so much, Michelle, and thanks to all the speakers. Before we go into our panel discussion, just a reminder to put your questions in the chat box, and we'll get the questions lined up. And you know, I think these collaborations that we've been able to form over the course of the pandemic between University of Toronto, University of British Columbia, Canadian Blood Services, DRDC, SIMVAR, and Canadian Forces Health Services, we've demonstrated the ability to conduct groundbreaking research, from moonshots like trying to control non-compressible hemorrhage on the battlefield to, you know, real-time patient-centered interventions that have a meaningful going to have a meaningful impact on the pandemic. So, and everything in between. So I'll open the floor for starting for some questions. And the first question we had was from General Mark Bilodeau, and his question was, would vaccine-induced antibody plasma work as well as convalescent equivalent plasma? The question's for Dana.
Yeah, so I think the answer to that right now is that we really don't know for sure. We do know that people who have had COVID infection and then subsequently get vaccinated have a huge boost in their antibody titers, and their neutralizing antibody is very effective. So we've been, you know, certainly encouraging people who have had COVID infection to go ahead and get vaccinated. Because of the nature of the vaccines, most of them are targeted to the spike protein. And so we know that the response of an individual who's been vaccinated is to make a certain amount of neutralizing antibody, but it's probably not got the same level of antibody repertoire that comes when you've actually had a natural infection. So for the most part, the COVID convalescent plasma studies that have been done have all been done with plasma from people who actually had real infection.
Thanks, Dana. Yeah, and so I think there's definitely a signal that when your immune system encounters COVID-19, your immune system really ramps up and you know, producing all types of antibodies. And I think, you know, with the current COVID-19 vaccines, I think probably the immune system's just not as ramped up as when you have it. And I think certainly some of the patients we've seen here clinically, and I'm sure Michelle has seen them, that, you know, the patients, their immune systems, they can develop a cytokine storm and have this massive inflammatory response because of the overwhelming immune response. Got another question for here from Officer Cadet Aidan Daphne. The questions regarding Dr. Kastrup's presentation is, he's wondering if there are logistical challenges to getting counterflow out to the battlefield. For instance, it could be easily included in a medic's pack. Furthermore, how does this compare to the standard device uses? So looking at maybe both the, you know, the packing and also the potential for getting it out into a product that can be injected into the abdomen for non-compressible hemorrhage.
Yeah, thanks. For the gauze, we think it's relatively straightforward. So currently, the combat soldiers would have a combat gauze, and so we can switch. I hope someday we can switch counterflow gauze in for instead of combat gauze, at least for some in some cases. It does have a biologic in it, thrombin, so there it might need to be it might not have as long of a shelf life as combat gauze, but so far we're really encouraged by the stability data. For the abdominal product, yeah, we also think that could be used in far forward situations, and it is just a CO2 cartridge, and then another piece that's about the same size as a CO2 cartridge plus the tubing. So I think it could be used there. It will require training, and so we will. Those are issues that we definitely are working through, and we hope to be able to use it in those scenarios. Yeah, thanks.
You know, one of the logistical nice things about counterflow is it's a powder, and so it's very light to carry. So it's not going to increase the amount of weight anybody's carrying tremendously. And some other products that are now coming on the market are more like a jelly, so they're heavier to carry. I think we'll be looking at the temperature excursion data on these products to see, how well does it tolerate temperatures over 40 degrees in storage and these type of situations as well. I've got one other question here is from Ramesh Zacharias, saying, in any of our studies, did we see any differences in sex and gender differences in any of the results from the various interventions? And I could say from the hemorrhage studies, all the animals are female animals, but I think, have we seen any differences from the convalescent plasma data in terms of male, female antibody concentrations?
Not particularly. The antibody range is very wide, and the antibody titers stay high for, our median was, I think, 128 days, and then they start to wane. And it doesn't seem to have a significant gender association.
Okay. Michelle, in terms of recruitment, is there any gender differences in terms of recruitment, or was it pretty even spread?
A pretty even spread, actually, which is good news.
Yep. All right, and I think one thing I'll ask Henry is that the freeze-dried plasma product we've looked at, and how has it been handling? How have you felt it reconstitutes in stores in terms of your studies? How robust is it in terms of storing the product and the reconstitution, and any issues with that with the freeze-dried plasma product? You're on mute. You're still on mute. Okay, so just chime in, Henry, when you get that sorted out. I think the one important thing with the freeze-dried plasma product that we've been pushing on this for several years now, and the COVID-19 did slow down some of our research efforts. But I think the important thing is, I think we're really realizing within Canada is we need to have some self-sufficiency in terms of these products for our own national security processes. And if you think of blood products and like PPE and vaccine production capability, we need to be able to have some resources to produce these own things with their own national boundaries for our own self-sufficiency. So these are some of the things that we've been looking at, and I think I hope that everybody got a taste of what we've been working through during the pandemic, and I invite anybody who's interested in this type of research to get in touch with me, and you can contact the organizers of the conference, and they'll be happy to provide my contact information, and we'll be happy to have anybody who would like to be involved with this type of research work with us. And I'll just give one last round for any questions. Anything from the last words from the presenters? No? Okay, with that, I'll wrap up the Q&A process, and I'll leave it over for conclusion of the meeting.
Hi, everyone. To all our participants, thank you for joining us today. Lieutenant Colonel Andrew Beckett, thank you once again for moderating an outstanding webinar. To our presenters, we're grateful for your expertise on this emerging topic that will undeniably continue to grow. Thanks to all, and have a great day.